Welcome to the fourth live-streamed New South Wales Science and Research Breakfast Seminar of the year, necessitated by the ongoing social distancing restrictions in place due to COVID-19. I'd like to begin by acknowledging the Gadigal people of the Eora Nation, on which at least I am gathered today, and on the nations on which you're gathered too, and to recognise their elders past, present and emerging. Parliament House has recommended hosting live events, or recommenced, hosting live events, albeit on quite small numbers. We're hopeful that the next seminar on the 2nd of December will be able to be held in person. However, these seminars will also continue to be presented via live stream. If anyone must leave early, an edited version of today's talk will be available on our website later today, alongside videos of our previous seminars. An important note, this format has changed slightly for this seminar. You will now see a panel on the screen's right, which enables you to submit questions. I will read these questions to our speaker at the conclusion of the seminar. Now, the sharp-eyed among you will have noticed that the topic of this seminar has changed slightly from when it was confirmed earlier in 2020. Distinguished Professor Dayong from the University of Technology, Sydney, was to give an address entitled Harnessing Light for a Healthcare Revolution. The ensuing pandemic has had an impact on Dayong's work, and he has focused today's talk on his recent research in developing a rapid, at-home saliva test for the early detection of coronavirus antigens in asymptomatic patients. Hence, the rather more specific and exciting title, Harnessing Nanophotonics for Early Detection of Coronavirus. Dayong is at the forefront of the fight against cancer and other diseases through his work on imaging and analytical devices. These devices use photonics technologies to analyze saliva, urine, or blood to identify early signs of diseases and toxins. He has developed a new family of nanophotonic probes called superdots, which can upconvert infrared photons into intense visible light at the nanoscale, with applications for biodiscoveries, data storage, anti-counterfeiting, super-resolution microscopy, nanoscale thermography, and recently, optical tweezers. Dayong established the Australian Research Council Industry Transformation Research Hub for integrating devices for end-user analysis at low levels, ARC Ideal Research Hub, and the Department of Industry, Science, Energy and Resources Australia-China Joint Research Centre for point-of-care testing. Dayong received the 2015 Eureka Prize for Excellence in Interdisciplinary Scientific Research, the 2017 Australia Academy of Science Engineering Science Medal, and the Prime Minister's Prize for Science, Physical Sciences of the Year 2017. Please join me in welcoming Distinguished Professor Dayong from the University of Technology, Sydney, as he presents Harnessing Nanophotonics for the Early Detection of Coronavirus. Thank you. Over to you, Dayong.
Thank you, Hugh. Thank you, Hugh, for the nice introduction. And good morning, everyone. I'm so pleased to speak at this highly prestigious seminar series. As introduced by Hugh, this seminar, when the seminar was scheduled early this year, I think none of us had ever imagined, how this pandemic was coming that badly. And since the first lockdown, my research program at UTS have been reshaped, and our team has been shifted our focus to developing the rapid antigen test. I think this is why I changed the topic of the today talk in reflection to our recent research activity and research programs. So I would like to take this opportunity to report our recent exciting progress and the very promising outcomes. Now, this is what our work looks like. You probably heard by this week, the total infection of coronavirus has exceeded 40 million people, and more than 1.1 million deaths. So this is a very bad situation today. So COVID-19 is now dominating our world with daily cases exceeding 400,000 a day, so nearly 20,000 every hour. So these numbers are still growing. Rather than seeing the peak, we all witnessed the first wave, the second wave, and a lot of countries are now experiencing their third wave. The third wave is going to be a lot of stronger and bigger. Okay, so the key challenge for this virus is they got very long incubation period, typically about 5 to 6 days. In some cases, can be up to 2 weeks, and that's why we require 14 days hotel quarantine, and that's actually causing there's a long incubation period actually causing a problem for contact tracing. Okay, so this actually sets the key challenge for the rapid detection of the virus. Now we have to refocus our research activities to see how we can contribute to this war, to this virus. And this is not what we choose to do, what we like to do. As a researcher, this is a time for us to plan and actually taking the actions on what we need to do and help resolve this terrible situation. Now here is the outline of my talk today. First of all, I want to quickly go through the review of recent news about rapid antigen tests. And then I'll show you, illustrate how this, point of risk testing window works and where is the opportunity and how challenging they are. And then a couple of technology being used, including the commercial product today, including the European probes and nanoparticles being used in antigen test, and also our recent development of antigen test 2.0 using the Nanophotonics upconversion superdots. And there are a lot of interesting and fascinating properties from this new family of molecular probe. They can even detect a single molecule or single antigen or single virus. I'll also show you some, little bit our research on the related area today, including the single molecule tracking in the live cell and also living organs. Of course, without a proper commercialization pathway, and these efforts, can be end up with, nice publications and in journals, in textbooks. And this is something we don't want to see. Okay, we want to see further of this research translation into industry. And by the end of my talk, I'll introduce our progress in collaborating with industry partner in Australia and where is the opportunity for our manufacturer and the economy. Okay, here is the piece of news published by Nature in 24th of September. It's saying fast coronavirus testing are coming. Okay, and this is the picture also shows President Trump holding this device. It's an antigen test device. The U.S. government recently spent about 760 million and purchasing about 150 million tests on the COVID antigen tests. Each test costs about $5 and the results can be returned in about 15 to 20 minutes. So this is amazing news, okay? So everybody, very excited about this news. Now the reality is, these tests, only get approval by the FDA for the emergency use authorization, okay? So that means this test is still premature, okay? So also FDA recommend this test to be used for people who already got the symptom and for the first 7 days after the onset of the symptom. Okay, so this is actually limit the window when this test can be used. But the reality is a lot of off-label use incorrectly, and that actually to try to screen people who are not showing the signs of any illness. But that could be also linked to the recent White House hotspot. So this explains actually the risk when a relatively low sensitivity test being used and miss the false positive visitors. And false negative could also lead to the some reckless behaviors by people who don't know they actually are sick. Okay. 2 days later, our TGA also approved 4 antigen tests to be imported from international, overseas. one from Korea, 3 from United States, but they all nasal swab-based test. Okay, so this is something still not so ideal. And also, this is what's being recommended on the TGA website. These tests are less accurate and only for symptomatic patients. And if you got negative results, you still require a confirmation test by PCR. But the PCR test is more expensive, typically costs about $100 per test. And the most difficult thing is for PCR, it takes about a day or 2 to get the results. So this is definitely not acceptable to handle this current pandemic. And later on, quite a few opinions been published. For example, this one published by Financial Review, saying, can we live with this virus with actually less accurate test? I really doubt about this strategy. Probably this strategy works for the normal, influenza virus, definitely not for this COVID-19, because this virus is so smart. It will take a few days for them to multiply within our body before our body shows any, response as a symptom. Okay, so the SARS virus will take a few days for them to find a preferred cell because they want to find a host cell so that they can multiply themselves. And then once they find the cell, and they will take about 10 minutes to enter the cell and a couple of hours, and for the single virus become a burst of thousands of virus. So this actually can be very dangerous when you test a patient who already get infection but only has very low amount of viral load. So the test will show negative. But this type of this group of patients, they still will take a few days to become a highly infectious in populations, and that could be a very risky population. Okay, so a lot of research recently shows people who could quite a large proportion of the people could never develop symptoms. And a mass test done in California recently shows about 53% of infected patients, they are asymptomatic. So these pre-symptomatic people still transmit the virus at extremely high rate to the people around him or her. So this is the symptomatic patient and non-symptomatic patient, they probably transfer this virus at the same rate. Now this figure also published by Nature recently shows the difference between the 3 possible way to detect the for COVID-19 diagnostics. You probably remember at the beginning of the pandemic, everybody rushed to develop this antibody test. From the news, it shows like a $5 test. You can return the results within 5 minutes. But later on, everybody realized this antibody test can be too late. So you can only detect the antibody in the patient's blood, as a response of our immune system a week after the symptom. That could be like 3 weeks once the patient gets the infection, gets the first exposure to the virus. That could be too late. The current gold standard PCR test using the nucleic acid, directly detecting nucleic acid from the virus, but that can explain that take longer for the biochemistry to amplify signal. And the recent rapid antigen test that directly targeting the fragment of the virus, and that's why this could be early detection. But this figure is still a little bit misleading, and that's why I redraw this figure to exactly show what exactly happened once a patient got the exposure to this virus. So in the beginning of this infection, as I show, this virus has become very smart. They keep in the silent mode. They try to find the best host cell in our respiratory system, and this will take a day or 2, and sometimes 3 days for them to settle down and find a cell. So this period is actually safe because the patient that doesn't have enough virus to transmit to other people. Okay, but the problem for this virus is once they enter the cell, they can duplicate, they can multiply themselves from 1 to 1,000, from 1,000 to 1,000,000. Okay, so the number will quickly climb up, and our body still doesn't show any response to this rapid duplication process. So this period is actually quite infectious. Okay, And before our body show the symptom, and there's a very large window, you can see the difference between the latency period and the incubation period. So this is exactly the window for point risk detection. The current antibody test, as I already explained, they cover the period, after 1 week, on the onset of the symptom. And the current antigen test actually detecting as a confirmation test being recommended by FDA, TGA, but only can be used for confirm a patient who already shows some symptom. Okay, so this is a detecting window. And the next window, which is if you want to detect this disease much earlier, you need to push down the detection limit, the limit of detection, so that you can push forward this detection window much earlier, a few days before the symptom. And this is the antigen test, next generation antigen test. We are one of the group try to using our unique solution for this problem. So this is a highly risky window for the point of risk detection. So this figure also shows, how difficult to detect this the presence of a single virus, coronavirus. At the beginning of the pandemic, I remember, that our public still, get confused between the difference of a bacteria and a virus. But from this figure, you see, it's much harder to detect virus than the bacteria because they are 1,000 times smaller in volume. Okay, direct detecting this virus, can be has basically 2 pathways. So this figure shows you, how possible we could detect the presence of a virus directly. So first method, as I said, this is a nucleic acid method. So each virus, they have 1 RNA molecule, and you can break down this virus, and you can use a biochemistry to amplify the RNA molecule. The antigen test is basically targeting the proteins on the surface of the virus or inside of the virus. So there are 2 typical, virus antigen we've been targeting. So one is called the spike protein. One virus, fortunately, they got 100 copy of this protein. And also, the current antigen test, they basically work on the nuclear proteins, which one virus you have about 1,000 copies. So this allows, better sensitivity to detect them. Okay. The step to detect this virus. First of all, we need to break down the virus. This is exactly, why we need soap to clean our hand, because in the soap, they got soap molecule, and on the soap molecule got the hydrophilic head and hydrophobic tail. And this hydrophobic tail can insert into this membrane protein of the virus so that they can break down this virus. How long that will take? A few seconds. And that's why we recommend the public using soap to wash our hand for about 20 seconds. Okay, so this is exactly the process these days we used for the virus detection, including this PCR test. Now this figure explains you why PCR test, the gold standard PCR test being used, and why this is so sensitive. Because you can use a biochemistry to multiply copy from the original copy of the nucleic acid and make an identical copy, and that's take about 3 minutes. But the first couple of cycles, they are not enough for us to detect them, and that's why we need to repeat this cycle for about 30 to 50 to 40 cycles, and until you can multiply from 1 nucleic acid into a million, a trillion to a trillion molecules, you can detect them. But this is a long process, and also explain why the PCR test takes so long to do, okay, typically by the professionals, health professionals. Also, in order to get a good testing results and sensitivity, these days we require the nasal swab, testing, because you can collect more virus from our nose. Okay, so this is definitely we don't recommend you to try this at home because it is not comfortable and it's very difficult to get to the right collection site. Okay, and that's basically the challenge for the current home-based testing. Why this is still not possible. In terms of detecting the antigen, there are 2 typical antigen proteins being recommended by the WHO. One is a spike protein, so they are on outside this membrane. Okay, they're like a spike, beautiful spike. They are more specific to the SARS-CoV-2. And the other type is nuclear protein. They are inside the virus. So as we already repeat this number, you got 100 copies of spike and about 1,000 copies of a nuclear protein from the per single virus. And this is good news, okay, than the nucleic acid. But the bad news is you don't have any biochemistry to multiply the number of proteins like you do with the nucleic acid. So that's set the challenge of why detecting this protein so difficult. The other challenge is the number of virus in our saliva. Okay, of course, directly collecting the sample from saliva will be more comfortable and non-invasive, and that will be more welcomed by our population. Okay, but they have slightly less virus in our saliva than from the nasal swab. Okay, but this figure actually very useful. I copied that from a journal publication recently published by a group in Korea at the beginning of the pandemic. That shows the level of a virus load in our saliva per ml, okay, per milliliter. Okay, so if you want to detect the presence of virus from the saliva, when the patient just show the symptom, you only have about 100,000 copies. Okay, and that's in that level you require a limit of detection about from 1 picogram to about 10 picogram protein levels. Okay, so this is a set of the challenge. And next, let's have a look how we could normally use to detect the antigen biomarker on the cell. So this is something we've been working building this research capacity for the last decade. In the beginning, we working with this company in Macquarie Park, Sydney-based Nanophotonics International. They are a biomarker company for cancer, prostate cancer diagnostics. So we've been working together for number of years. And in the beginning, we are using this European probe, and which is a fluorescent biomarker, and we can label directly to this antigen on the surface of a cancer cell. And remember, the cancer cell is a lot bigger than the virus itself. But this is show you the same principle and the capacity, and we can do this. Okay, and that allowed a background-free imaging of the prostate cancer cell in urine. Okay, this is some results we reported a few years ago. And once we have a low number of protein, biomarker, antigen biomarker on the surface of the cell, and we struggle with the signal strength and how we do it. And then we start to use the nanotechnology. And of course, this is in collaboration with material scientists in the beginning, and we learn how to encapsulate many signals in one nanoparticle. So once you have many signals in one nanoparticle, you will see, this is actually give you the chance to optically amplify your signal. So this is the standard process. We prepare European nanoparticles, and this is where the nanoparticle get excitation at UV, and you can check the beautiful red color. And because the long Stokes shift, and you can separate the excitation and the emission signal, and that's guarantee the very good sensitivity. And also you can put thousands or even 10,000 of European dye inside the nanoparticle, so that you can use this nanoparticle as an optical amplification signal to get a sufficient amount of the signal on the surface of the cell. And this figure shows you one of our early results as a collaboration with BD Biosciences in San Diego in United States. And that time we try to use our technology to resolve very low amount of antigens from a leukemia cancer cell. And this figure shows the difference, the contrast when you use a single dye and you use a smaller nanoparticle, which is about 40 nanometer, and you keep increasing the size to about 200 nanometer, and you can encapsulate more European signal, and that's why you can see the signal getting brighter and brighter. technology being used in the current antigen test commercialized in the market by a lot of companies. Okay, so this is even through this new technology using nanoparticle to amplify the signal and using this European dye, and still you have a quite limited detection limit. Okay, and that's why this figure shows you FDA only approved the antigen test, current antigen test as a confirmation test once the patient start to show the symptom. But the dream is really, the goal is really keep pushing the limit detection and see how can we detect this infection earlier than the symptom. Okay, so that's why we require a more sensitive antigen test. So now I want to show you a couple of ways to further enhance the sensitivity. And this is a technology we've been used in our lab, try to detect the Giardia using time gating. Time gating, like you have this probe which has a longer fluorescent lifetime, and you can use a pulse illumination to difference the signal from the background. So this is exactly like the scenario when we're watching TV at home, and suddenly, in the evening, and suddenly the house gets powered off. Once you got powered off, everything else went to darkness suddenly, but your TV screen still shows some ghost image. So this is exactly the material, a similar material will be used as a fluorescent marker to label the single cell. And that's the strategy, you can see the ghost image in the darkness clearer. You have a better contrast, signal to contrast ratio. So this is a non-time gated, and this is time gated. So potentially, this is another technique to further improve the sensitivity. The other very exciting opportunity, which we have been developing, for the last few years, called upconversion superdots. So this is a new family of a material. As I said, I show you, normally fluorescents will be excited at shorter wavelengths because they have a higher energy, and you yield the fluorescence from longer wavelengths but lower energy. Okay, when you illuminate jellyfish by a UV or blue lamp, you can see beautiful color of green. Okay, so this is a normal fluorescent process. This material is very unique in terms of the photonics. So you can they only absorb the emission from near infrared. Okay, so this is very low energy photons from infrared, near infrared. And then you can combine multiple near infrared photons into the visibles. Okay, and that offers you the opportunity to detect the signal from darkness, because the near infrared only excites the signal material. And this is sort of material we've been spending a lot of time in the last 5 to 6 years. And we said to myself, my team, we got addicted to this science. And later on, we get a lot of new properties of this material, and we publish a lot of beautiful papers in the literature. And we also try a number of ways. So this is really the central focus of our research. Try a number of ways to keep enhancing the brightness of the signal by giving high number of excitation, increasing the quantum efficiency, and most importantly, we try to enrich the emitter, the number of emitters per nanocrystal, from 10 of them to hundreds of them, and now we can put thousands of emitters per nanocrystal. As I explained to you earlier, and this is an effect we can optically amplify the signal from a single crystal. So this was really, starting from a fundamental research, and also as a result of a collaboration with the team at University of Adelaide, with Professor Tanya Monroe's team, a few years ago, and we published these results 7 years ago, and we name it later on as a super dots. Nature, they call this a super bright nanocrystal, and that's the first time we realized the single nanocrystal sensitivity. And you can see this is a normal doping. They give a limited signal, and this is our highly doped material actually can enhance the signal by up to 2 order magnitude, and enhance the detection limit by 3 order magnitude for single molecule detection. So this is very exciting. After that exciting research, we're expanding our research activities around this area, including getting this material emitting brighter signal, higher quantum efficiency, control the size and shape, and also control the color. So not only from the green, also from blue and red, other color, and also a lot of barcodes in time domain, polarization domain, and that make this crystal can be useful for a number of applications, including the anti-counterfeiting applications. But this is not so relevant to the talk I'm going to give today. And this one is a very exciting research we conducted together with Mnemonic International at Macquarie Park and by my student, Hao He, during his PhD. So we published these results early the year of 2018. That's more than 2 years ago. So the research is about, we translate this bright material for strip testing, and we engineer quite small handheld device, which you can put on a smartphone, and you can read the signal and sending the testing results to the cloud. And this is the internal design of this material, and you can see this is exactly the same lateral flow strip we use for pregnancy test, but we change the reagents. So instead of using the gold nanoparticle, we're using the up conversion superdots. You got two color, one is yellow, one is a violet-ish color. So you can use this two color to recognize two cancer biomarkers simultaneously, and this is suitable for home-based testing of prostate cancer patient sample from urine, for example. When the pandemic came, we thought, among our number of technologies, and this is probably the most easy, lower hanging fruit we can use for pandemic, for this COVID-19 detection. And this is exactly the design of our strip. We have two color, okay? We have two testing lines, and one is for spike protein, one is for nuclear protein. And the third one is a control line to illustrate whether the device is working or not. And very excitingly, my team got these results last week, and I'm allowed to show this limit of detection. So this figure shows, we now really challenging pushing the limit detection from 100 picogram to 10 picogram and now to 1 picogram, okay, per mil for spike antigen. And with the sensitivity at between 80% and now pushing to 90% level, okay. So this is a very exciting preliminary result. Remind you, this is only about 2 months. After 2 months, hard try in the lab by using the new antibody and antigen for the validation. And we prepare to do more large-scale testing, in our lab, also in the PC3 lab environment before the Christmas, definitely. And this is something I want to show you why there are plenty of room at the bottom, because you can see the limit of detection. We're never happy about the limit of detection because we always want to detect this disease much earlier from the patient who showed the symptom or few days before the symptom. Or can we detect this virus once we just get infected, just get exposed to the virus? Okay, we call it single antigen detection, whether this is possible. I'll show you how possible our technology already demonstrate. Okay, so this is the work we published 2 years ago. We built a simple microscope. Okay, so in terms of detecting a single molecule, you need at least a simple fluorescent microscope. Okay, purpose built. So this shows you, we can now, using our naked eye, directly see these single molecules under a microscope. How much photon we need? Okay, so this is a figure shows you, how much photon we need for our eye to directly see these single dots. And you see, this is a, this material, they are highly power dependent. That means the higher power you apply to this material, the brighter signal you will get. But at a certain level, it's around, from 1,000 to 4,000 photons per nanocrystal. Our eye can directly see these single dots through a microscope. And the next result shows you, this is sometime, when I show this to biologists, they are very excited about these results because this shows you a single cell under a normal transmission microscope, and you add a up conversion laser, non-AT near infrared laser, and you can see actually the interaction between a single nanoparticle and a single cell by our naked eye in real time. So this is a very handy tool for the biologists to understand how the nanoparticle come into the cell and maybe as a drug carrier to carry the cell, the drugs into the cell plasma and cell nuclear. Okay. And as I said, before the pandemic, we've been always, highly motivated for the early stage cancer diagnostics. And this is some preliminary results we've been done together with Myonomic International at Macquarie Park. And we use a standard, Petri dish and 96 well. And we can do the standard ELISA assay, but the detection and the reagent we use for this assay is different. We use superdots, which can sandwich the prostate cancer antigen on the Petri dish, so that we could, using our super resolution imaging microscope to identify the single molecules. We can quantify the very low abundant single antigen molecules from the patient sample. Okay, this is a very preliminary results. And the story doesn't end here, because there's a fascinating development of nanomaterial engineering. And this is the diagram we achieved 4 years ago in collaboration with the National University of Singapore and the University of Wollongong. We demonstrate actually these dots, they are smaller than 50 nanometer, definitely smaller than the size of the virus. And we can purposely engineer a single dot with multiple functions from a spherical shape to non-spherical shape. And this is going to be very useful for the clinicians and the doctors use as a multimodality imaging contrast agent. You can simultaneously combine the optical modality, magnetic properties, and also the X-ray properties for multi-resolution and multimodality imaging in the same time. And here is a very interesting result we collected, 3 years ago as a collaboration with scientist Professor Sean Jackson and his group at Heart Research Institute in UC. When you see this map, you probably thought, oh, this is probably Australia. I'll tell you this is not Australia. You can see the scale bar is only 2 microns. So this is a platelet cell under a microscope. So normally the platelet cell, they are small and our microscope doesn't show sufficient resolution, and we start to use super resolution to see the single molecules on the platelet cell, particularly when the cell are alive. Okay, we want to watch the behavior of the cell. And more advanced vision for this research is we want to send our nanosensors, nanophotonic sensors into the cell, and we want to locally detect the localized temperature, the pH value, the force, and also the traffic condition within a single cell. So that's become very possible for us to build this street view version of map from a single live cell. Another very exciting collaboration with Professor Martina Standil at UNSW, and her group is more interested in the research developing new chemistry and new nanomedicine and deliver these drugs into the cancer cell, in particular into the mini organs, try to understand the drug response and pathways. But the current limitation is about resolution, is about microscopy. So we can use these super dots for this application, because on this figure, it shows the cluster of billions of cancer cells. They form together a spheroid. This is a model of the mini organ. But the problem is that the current microscope doesn't see that deep. Okay, so I can use an illustration here. So today I'm using this laser pointer is red. If I'm shining the light from one side of my finger, you probably can see it from the other side. This red light, they have a good penetration through our skin and tissue. Okay, but if you use a green laser pointer, you won't see much light can be delivered. So that means our tissue has a window closer to the near infrared range. So once you have a red and near infrared range, the light can be penetrate much deeper. So this is exactly the technique we use for this spheroid mapping of single nanoparticles. And because nanoparticle itself get excitation from near infrared, and they can emit near infrared emission as well. And the other one is very exciting as well. We showed these results and published 2 years ago as a collaboration with Fudan University in China. You can see, these dots, once you inject into the living mice, And after 24 hours, these dots can recognize the size of a tumor, and you can this one, it will potentially be very useful to guide the imaging-based surgery for our clinicians. So this is a very exciting. Okay, without a good commercial pathways, our project can be end up with a paper only. We want lots of more out of this. So 5 years ago, we set up a industry and university collaboration consortium. We called, Australian Research Council funded research hub for IDEO with, 6 industry partners and also our partner university, University of South Australia in Adelaide. And that's including some stories I already told you today about cancer diagnostics, about illicit drug sensing. Okay, and that's later on, 2 years ago, we set up another entity. It's taking the funding jointly from Australia and China for this joint research center for point of care testing. Again, more Australian industry have a chance to join this consortium for this commonly interested area for point of care testing, including SpeedX DNI Design Company and the University of Wollongong. And this is a very exciting story. Starting the mid of this year, although we had a collaboration with Alkalizer in Perth for a number of years, and we've been working together within the hub, within the center, and for the problem of illicit drug and alcohol test on the roadside. Once the pandemic come, in the first instance, the company quickly switched their focus to increase their manufacture capacity, making these test strips about 20,000 test strips a day now. They receive federation government funding, and the company invest a lot into these assets. And this is in the beginning, we study how to making more test strips for antibody tests, and now the company keep pushing the limited detection and the technology by collaborating with universities, with UTS, with South Australia, and we now start to prepare our manufacture by this robotic machine and commercialize, upscale the production of this antigen test strips. So this is some milestones we already achieved, and also the planned milestones in the next few months before these products can be in the market, can be used in the airport and in nursing homes, for example. If you have, if you're interested, you can follow the website either from UTS or from Alkalizer's website, and to see our fresh progress along this pathway. Now, before I want to end up this talk today, I think my time is nearly there. I want to share a short video. Okay, this will give you a summary of the technology we've been developing today. Scott, can you do me a favor?
So why this virus is so contagious? Because our body is very slow in response to the infection. It typically takes a week before our bodies start to show the symptom. The goal for us is to use a nanophotonics approach to detect this window, okay, before the symptom, but they're already infectious.
The test mainly based on the nanoprobe technologies. These probes, they are super bright, and their signal can be sensitive to the single molecular level. After adding the saliva on the strip sensor, we can read the optical signal by using a meter, so that the proteins of virus can be detected.
Unlike current conventional tests that take the swab and takes several days to get your results, our test can deliver that result within minutes. So we can detect asymptomatic and symptomatic people around. So for example, in the airport, if you decided to travel, you can get tested within minutes. And if you're negative, you can continue with your planned travel and enjoy your vacation. It's Australian innovation, and together with Australian company Alkalizer, we can create rapid tests for COVID-19 here in Australia.
All right. So I hope you hear the sound from my side. I couldn't my end. I couldn't hear the sound. But anyway, you can have access to this video on UTS website. Okay, so this is a conclusion of my talk. It's a bit wordy, but I still want to emphasize, this long incubation time of COVID-19 requires us to keep pushing the limit of detection from picogram per mil to femtogram per mil range and identify the asymptomatic patient individuals. Time domain and upconversion nanophotonics offer a great opportunity for the test become a single molecule sensitive on the antigen test. And also we emphasize the sustainable university and industry collaborations, and they are very essential pathways for industry translations. We now urgently need the access to PC3 lab environment and the patient sample so that we could validate our test in larger scale. We want to build, the research capacity in the area of photonics and nanomaterials, and they are basically the key to disruptive biotechnology technologies. Okay, and the key I really want to emphasize is to build multidisciplinary international and industry collaborations. Finally, I want to thank our founding agent and a lot of funding in the last decade, and also my team at UTS and a lot of collaborators across Australia and internationally. Thank you so much.
Dayong, thank you so much for a fascinating talk. We've got a complicated process now where I've got to keep an eye on my emails for questions, so just bear with me. But I might start with a couple of my own, actually, if that's okay. I mean, it strikes me that you've got a tool here where you can detect anything almost at the individual molecule level. And I'm just wondering what the limitations are, because I have in mind, things like, I don't know, explosives detection or other molecules other than organic molecules. Does it work for that? Or does it require growth or what?
Yeah, so the reason I call this a super dots, I really wanted this material become super capable for everything. But reality is, depends on how you design these sensors. So I didn't cover this today in my talk. This sensor recently become one of the interest for thermometry, thermometry test. You can put the cell, put a single particle into the cell. You can detect the localized temperature generating from mitochondria, for example. And also you can use this material for pH sensing, okay? Also for gas sensing, but depends on how we are using different physical properties from this material for real world application. And the most exciting one also include the one we recently discovered. This single nanodots actually can generate a relatively huge force under the optical tweezer, so that we can use these dots as a force tag to measure the mechanics properties of biomedical, biomedical substance. So the focus for us has been more focusing on the biomedical technology, but of course there's a lot of other work done by the colleagues internationally around this material and to investigate this material for lots of broad applications.
Yeah, it sounds like you could also use it for, I guess, an alternative form of microscopy, and things like that. Yeah.
Yeah. So we spent a lot of efforts on microscopy recently. Yeah.
Yeah, which I kind of, I have a lot of interest, and the state has a lot of interest in how we better commercialize our research. I'm sure you're aware. You talked a little bit about the companies that you work with, but have you? It sounds to me like you should be thinking of lots of startup companies and things like that. What are you doing in the kind of startup area? We have the physical, things like medical devices fund. Yeah.
Yes. So we try a number of times for medical device fund, but the outcome wasn't so good because very competitive program. I think we still shortage of funding in this area, and that's the key. And in one of my recent comments, I said, although we know this is very important to, sort of reshaping all the efforts for the COVID-19, but still, including COVID-19 problem, still we are shortage of funding. We know this is important, but the embarrassment is we shortage of funding. That's a reality, okay? And startup company also struggling in Australia, we all know, and we directly working with, about a dozen of startup companies from SMEs in Australia, including the ones I already told you the story today. But they all struggling, because the environment and also the opportunities, international opportunities. So we definitely need a lot of more work. But this is can be joint efforts between universities, between the companies, and also importantly, the government. How we push this in a very sustainable way to get this happening. But it will happen. It will happen. And from the journey I show you, also we learn a lot of experience. We train our students to become more adaptive in the industry environment, and some of them already pick up jobs in the industry. You can see the change, but I will say still a lot more we need to be done.
Yeah, look, I agree with you. I have to say, I think you and I need a follow-up conversation because I'd be very interested in seeing what we can do to help you commercialize things from a government perspective. I don't know quite how the questioning system is working, but I've had no questions come through at all. So I have a suspicion that the new system is not working properly. So I'm sorry about that, Dayong. But what I will say is, look, if people do have questions, I will forward them on to Dayong and get him to answer those. And I want to thank Dayong again for a fantastic talk, really, really interesting, and definitely different from anything that I've ever thought about or heard about before. So really great stuff indeed. So I'd just like to conclude.
Thank you, everybody.
Yeah. I'd just like to conclude. I think Dayong received a wine and a book in the traditional manner for the seminar.
Oh, I did actually last night. Yeah.
Okay.
This is the wine I received last night. Thank you. Thank you, Hugh.
The chief scientist Christmas hamper works. That's good. And also to remind the audience to join us on Wednesday, the 2nd of December, when distinguished Professor Antoine Van Jogen from the University of Wollongong will present Tackling Antimicrobial Resistance from Molecule to Community. And I gather Antoine is actually a good collaborator of yours. So that's great.
Yes.
The office will be in touch both via its email database and Twitter to confirm whether this seminar can be held at Parliament House as well as presented via live stream. So thank you everyone for attending this morning. Much appreciated, and thank you again, Dayong. Bye.
Thank you, everyone. Have a good day.